Thank you very much for the nice introduction, and I would like to thank Prof. Pierce Chow for having me here to introduce our work in tumor evolution and how it correlates with prognosis. So this was my previous work in Genome Institute of Singapore. Currently, I'm working in Evolutionary, also working on using AI to decipher the genome and to understand how to benefit the patient more. So similarly, in this study, we try to link our basic study in cancer evolution to the clinical impact. So this is basically an extension of the study from Weiwei's previous presentation with a larger cohort. And here we are also using multi-sectoring sampling technology and whole genome sequencing and RNA-seq, but on a larger cohort of 127 patients with prospective survival follow-up data. So the drawback, the limitation of previous study is that they lack a framework to summarize from genomic evolution to transcriptomic phenotype to prognosis stratification. So here I would like to present to you some framework or principle of how you can unite multi-omics data together to understand the utility of evolution and heterogeneity in patient prognosis. So let's start with the genomic evolution paths. So first, a very short introduction to what are the evolutionary paths. It's basically the path that a tumor may take to develop, and this path is measured by the occurrence of driver events across time. For example, this is data from renal cell carcinoma from earlier Tracer X research by Charlie Swanton. So they developed a method to time the event, the occurrence of the event. For example, VHL gene mutation may happen earlier during tumor evolution, and then later followed by PBRM1, and later followed by SETD2. So this is one path a tumor could take to develop, and there may be other paths within a cohort. So by identifying these different paths, maybe we can understand how different tumors would evolve under different circumstances. And actually, in those previous studies, they also find that this kind of evolutionary path will result in quite different patient prognosis results. And it's not restricted to renal cell carcinoma. It's also shown in breast cancer and also non-small cell lung cancer. So now the question we have here is whether we can identify similar evolutionary paths in HCC and whether it could affect clinical trajectory of HCC. And answering this question might be very helpful to answer some of the previous questions Weiwei raised and the fraud raised to make use of the evolutionary data we have from such a big cohort. So we begin with using the same method to stratify, to segregate the patients of our PANI cohort into different evolutionary paths. So my apologies that I cannot show all the details since this is the work unpublished yet. But basically what we have done is we try to use the same method, and based on the timing or clonality of the mutations, we separate patients into different subtypes as shown here. Each column is a patient, each row is a event. For example, this is one of a very dominant event occurring in 3 of the evolutionary paths. And then for this path, they actually show a dual mutation event for both gene A and gene C. And for this path, they have gene A and gene B event, and this is only gene A event. So by segregating patient in such a way, we ask whether this has prognostic effect or not. And this would be the starting point of our analysis. So that by showing the Kaplan-Meier plot here, we do see that 2 of the subtypes, which happens to be the dual mutation subtypes, show worse survival compared to the other subtypes. Although it seems it's not significant in this plot due to a slightly limited sample size, if we group these 2 subtypes together because both of them are dual mutated subtype containing the gene A, so we can see it actually segregate quite distinctly from the other subtypes. And actually such separation remains significant even when we control for stage, age, gender, and virus status. So here we conclude that actually there are recurring evolutionary subtypes in HCC, and they have the effect of prognosis. But then we ask, like, according to the central dogma, mutation is just the very first step, and whether such evolutionary subtype alter the transcriptomic landscape to confer these prognosis differences. So this leads us to our next section of the presentation about transcriptomic subtype and how it linked to the evolutionary origin. So to understand this, we first perform unsupervised clustering, and here we identify 6 subtypes, and concordant with various previous presentation in our subset of this cohort, we find a high proportion of the patient would contain mixed RNA subtypes within a tumor. And then we try to understand whether some of the subtypes, similar to various previous presentation, whether some of the subtype would show worse survival compared to the others in our prospective cohort. So to simplify the problem, we only focus on this pure set of patients first, and we do see that again 2 of the subtypes show worse survival compared to the others. And then it happens that these 2 of the subtype is very far away from the adjacent normal from this PCA plot, indicating that they might be more aggressive than the others. And now we call them bad apple subtype, and I will explain more later on why we call them bad apple. And so the next question is, we want to understand what are so special of these 2 subtypes, like what are the pathways that has changed in these 2 subtypes. So we perform a pathway analysis. So the frame here showing the bad apple subtypes, and we see that they seem to show lower expression in metabolism related pathways, but a higher expression level of proliferation or cell cycle related pathways, which indicate why they are more aggressive and tend to show bad prognosis. And also, we try to correlate our findings with previous publication, and these bad apple subtype were very similar to the subtypes that show bad prognosis in previous publications. So this looks like quite robust results. Then we ask whether it has anything to do with the evolutionary subtype we found before, and as you may guess, maybe these are enriched in the dual mutation subtype, which tend to show worse survival. So indeed, we observed this phenomena that in the dual mutated subtype, including the subtype that has a gene A mutation. Again, a reminder that all these 3 subtypes contains gene A mutation as a truncal early event. And among this set of patients, there are much higher proportion of tumors containing the bad apple subtype. And also we can view it from the other angle that among all the patients with bad apple subtype, more than 60% of them come from this 3 evolutionary pathway. So with this, we can draw a first conclusion that it seems like still the prognosis and the phenotypic landscape actually still follow the central dogma here, that how the tumor evolved from the genomic path could determine to some extent how the transcriptomic profile would be. And then that may affect, again, the prognosis of the patient. But here we left one question not answered, is the impact of heterogeneity, as shown previously by Wei, that heterogeneity in transcriptomic or even genomic profile could have some effect on the prognosis. And how can we integrate this level of data here in our analysis? So this leads us to the final session of my presentation about the bad apple principle for prognosis prediction. So as you may guess, like the more heterogeneous a tumor is, the worse survival the patient will be. So we try to test this hypothesis with a simple pure mixed segregation of the patient. So you can see here, actually, patients with mixed RNA subtype and those with pure RNA subtype actually don't segregate too much based on the recurrence-free survival. And moreover, if we use the RNA ITH, like the level of heterogeneity in the RNA expression, and we don't see a strong segregation effect in recurrence-free survival. So considering we find a very aggressive subtype that show worse survival in the previous analysis of transcriptomic, then we ask the question, for the patients with this bad apple subtype, whether the mixed patient will survive worse than those without, because they should already show very bad survival, then whether ITH, the transcriptomic ITH, would add another layer of stratification here. And with our larger cohort here, we can answer this question. So this is the result. We can segregate the whole cohort into 3 groups of patients. One is purely bad apple patients, like we only find bad apple subtype among this set of patients, and the mixed bad apple patients, which we have a mixture of bad apple and other subtypes in this set of patients, and all the others. And you can see a clear distinction, and it's a gradient of segregation of prognosis across all these layers of patients. So it seems that as long as you have the bad apple subtype in your tumor, then the patient will suffer from worse survival. And that's why we call this a bad apple effect, because there's a saying like one bad apple can spoil the whole barrel. So again, such segregation remains significant, even controlling for stage, age, gender, and marital status. So we showed before that mixed subtype shows slightly worse survival. Actually, it may be due to the fact that the mixed subtype patients would have a slightly higher proportion of bad apple compared to those with pure patients. So it seems that apple subtype is a more strong confounding factor in prognosis stratification. So again, to provide a better picture of how ITH works in patients' survival, we stratify a patient into those with bad apple and those without bad apple, and see how level of ITH can stratify patient here. And we see, very interestingly, it seems there's a double-edged role of transcriptomic ITH, in that the patient with bad apple, the lower the transcriptomic ITH, the worse the survival would be. But it's the opposite in the patient without bad apple. So how to understand that is like the bad apple is really driving the worst survival. Thus, if you have a lower transcriptomic ITH here, it means that almost all of your tumor parts are of bad apple profile. Thus, it tend to be much worse compared to those that contain some other non-bad apple subtypes. Whereas in patients without bad apple, the patients with higher heterogeneity might have higher chance to evade the immune system, or might migrate to other sites to have recurrence. So heterogeneity works very differently among patients with bad apple and without. And to demonstrate this point even further, we come up with a bad apple principle, that it is the most aggressive part of the tumor that has the most power in prognosis prediction. So to demonstrate this, we use another published prognostic biomarker, which is called the MS score, as shown in this paper. They use a MS score, a multi-gene expression-based biomarker, to predict whether patient will recur or not. So they have a very strong segregation in other cohorts. And using this score in our cohort, because we have multi-sectors, we can, for each tumor, we can use the minimum value of all the sectors for this MS score, or we can use the mean, or we can use the maximum value. So based on the cost survival analysis, we can clearly see that it is the maximum value across multiple sectors that can predict the patient prognosis using this published biomarker. So again, this to some extent confirm our hypothesis of the bad apple principle, that in order to have more accurate prediction of prognosis, we should choose the parts that show most aggressive transcriptomic profile. So as a summary to this tiny story is that we identify several HCC evolutionary paths that can stratify recurrence-free survival, and such paths actually can predict the occurrence of a bad apple subtype which can predict the patient's survival in a apple principle manner, that the most aggressive part has the most power. So yeah, with that, I would like to end my presentation with acknowledgement to Prof. Wei Wei and Prof. P.S. Chow and the team, including Naslihan and Wen Huan and Xin Qi and Lin Yan, who's joining here also, among others, who has worked tremendously hard on this and provide a lot of data and interesting insight. And finally, thank you all for your attention, and I'm happy to take any question from now. Thank you.